Good morning, everybody. My name is Barbara Abrams, and I'm going to be moderating the next session. The title of this session is The Dual Burden of Malnutrition and Obesity on Global Health, from Infections to Chronic Disease. And just as a bit of introduction, the idea of this session was to take such a comprehensive approach and to go from literally molecular to highest level policy. So we're not sure how that will work, but the two speakers are going to valiantly tackle this. So our first speaker is Dr. Mark Manary. Years ago, I was inspired by published research that addressed a really intractable problem in global health, which is when children are severely malnourished, rehabilitating the children to allow them to grow and thrive and essentially gain the weight they need and establish the nutritional status that they need to, to survive and to thrive was a very difficult problem. We weren't very good at it in the first place, and when we did it, people were put in hospitals and separated from their families and communities. So when I read this new approach, which was using a peanut butter product that was very, very easy to deliver and therefore could be used in the community and even in the children's own home. The product I originally read about was Plumpy Nut. I realized that Dr. Manary was one of the leaders in this field, and he has remained a leader in this field, spending a lot of his time in Africa and also in the United States publishing and teaching in the area of understanding the underlying mechanisms that lead to child undernutrition, as well as interventions to cure child undernutrition. And this peanut butter product is really, really effective. He has now developed his own NGO, which is known as Project Peanut Butter, and I don't know how much of that he's going to talk about today, but I did want to highlight his website because it's really, really a great website where he describes the work that he's doing there. He works in Malawi, Sierra Leone, and more recently in Ghana. And my assistant, who was an undergrad here at Cal, told me that Cal has run a race to raise money for your organization. So, yeah, I just found that out. So all of that is just background for the real purpose of his talk today, which is when we began to plan this conference, it was really clear that one of the topics that is really groundbreaking in the field, really possibly shifting a paradigm in the field of nutrition and health, is the whole understanding of the human microbiome. And again, Dr. Manary's name popped up. And so we invited him to come and speak today on microbiomes and nutrition. So I'm really pleased to welcome him.
Great. So thank you for the kind introduction, Barbara. And likewise, when I got the invitation to come, I was kind of excited. There's a lot of great science and scientists going on here. And then asked to talk about the microbiome. So I thought, okay. Well, a little bit more personal sort of background. I'm a doctor. I'm a pediatrician. I've been working in Africa the better part of 28 years. Although I may be paid by Washington University, they define my job as 11 months a year Africa. And so I'm a very earthy, hands-on sort of clinician. But I believe that... I believe in science and knowledge that these have some basis beyond and power beyond opinion. I think that biology will trump any amount of policy or advocacy that's falsely based. And I also don't think that I should be limited by what I don't know. So try to learn lots of new things. So what I want to do is weave a story in about how I got interested in the microbiome and what it might mean in sort of a pretty big picture way. Now, how many of you have heard of the microbiome? I assume everybody. Everybody's going to raise their hand. Because this has been, like space travel was talked about in the late 1960s. I mean, everybody's heard about the microbiome, its potential, its power. But not much has happened to deliver any benefits to humans or human health really yet. The problem that caught me is this child here. So this child here, was appropriately breastfed for 21 months, and then they stopped breastfeeding, then they had a couple episodes of malaria, and then they come to a village health shelter like this, seeking help. The child's swollen. If you can hear the child, they're groaning continuously. They're anorexic. They're irritable. And they're a bit underweight. And this problem, that you probably all know, is severe malnutrition. It's kwashiorkor. It's one form of severe malnutrition. And I didn't really encounter this in such a big way until I went to Malawi. In fact, I'd worked in Tanzania, I'd worked in West Africa, I'd worked in New Guinea. But when I got to Malawi, I said, wow, the problem here is kids are sick because they don't get enough to eat. And there were lots and lots and lots, probably 30 or 40,000 kids a year in Malawi with this kind of problem. And it's a fascinating thing. If you're a doctor, what is causing this problem? It's a very serious thing that these mothers is not lost on them one bit. They realize that if this kid doesn't get something appropriate therapy, in a month they have a 2/3 chance of dying. So, as Barbara pointed out, we went and developed a food therapy, which is very effective at treating these kids, but wondered in the back of my mind, how could we prevent this problem? I mean, can we do something on it's a public health problem there. We're talking about 2% of all 1 to 3-year-olds have this kind of swelling. And what's the deal there? So I thought about it. You read about it. You understand that the idea of protein deficiency went out with 1980, and there's good evidence that nobody should think this is about protein. And there was a hypothesis about antioxidant stress. And so we did a randomized double-blind placebo-controlled trial of a cocktail of antioxidants in a couple thousand kids, and it didn't prevent kwashiorkor one bit. We did prospective dietary work where we categorized how much and what foods these folks ate, these kids who were coming up to hungry season, who we knew that were vulnerable to developing kwashiorkor, and the children that got kwashiorkor didn't eat anything different than the children who did. Not in quantity, not in nutrients, not in foods. So, something in the environment, we don't see this around the U.S., kwashiorkor, but what is it? So I started to think about, and this would be sort of in 2005 or so, what in the environment could be causing such a problem for these kids that we can't really detect. And I'd heard about the microbiome too then, and one of my colleagues who apparently was just here a couple days ago, Jeff Gordon, was actively working in the microbiome, so I began bugging him about, well, can we look at this microbiome thing in kwashiorkor? At that time, the ability to sequence deeply was not nearly as strong as it is today. And so I had to bother him for several years because at that point in time, 3 samples is sort of a graduate student's 8-month project. But when the ability to analyze hundreds of samples came on board, then we undertook a project about kwashiorkor. But let me make a few comments here about the microbiome as a way of background. So, what this is all the microorganisms that are on the body surface. But really what it is talking about the gut, okay? Because almost all of these organisms are in the gut. 80% or so are in the colon, about 20% are in the small bowel, and then if you compare the numbers on the skin and the mouth and so forth, they're very, very trivial compared to those numbers. 100 trillion organisms. They involve bacteria, parasites, yeast, a whole diversity of a population. And the number of genes that this population, the microbiome, has is about 100-fold greater than all the genes in our body that are active. So the genetic diversity and the genetic capacity is huge from this what is sometimes referred to as the 6th organ system. What does it do? Now we'll talk a fair bit about nutrition or allude to that more, but you should keep in the back of your mind we don't really know a lot about what this does at all. So I think the story, if you go 10 years ahead, about it being very integral to the immune system is going to be much different. But it extracts energy from food, sort of fiber, non-digestible carbohydrate. It creates or synthesizes short-chain fatty acids, which feed some of the important cells in our gut and also provide a immunomodulating effect. Provides lots of enzymatic reactions that we need. For those of you who know about nutrition, you probably know there's like essential amino acids and non-essential ones, and vitamins are things that we need to get in our diet because we can't synthesize them. These microorganisms can do it all, and that's a huge source, underappreciated, of all these vitamins and minerals and essential nutrients. I mean, vitamin K, I think, is kind of the poster child example. I mean, we don't make vitamin K. We need vitamin K, and it's all made in ample quantities by our gut flora. The microbiome also detoxifies, dietary toxins. So if we think about something like aflatoxin, having a competent microbiome reduces the carcinogenicity of aflatoxin about 5-fold. So it has a protective mechanism. And then in terms of the immune system, it's also very protective because, our gut interface, which is really where environment meets the body, is a thick layer of mucus, which is a physical impermeable layer, and then there's all kinds of antibodies and molecules, cells eking their way out there that kind of provide a defense system. And this microbiome is orchestrating that. It's calling for the macrophages to stay in one place and the T cells to move in another place. No, it's not sort of happenstance that, whatever you happen to have there, that's your microbiota. But there's a core, a reproducible core that we understand something about that contributes to human health. It can vary, of course. There's basically 4 large classes of bacteria: Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. And just in very broad strokes, some of these groups are more pathogenic or more inflammatory than others. Some of these groups are good at digesting animal source foods. Some of these groups are good at digesting plant source foods. So we need it. It's there. It's common to all of us, like many of our genes are common to humans. And certainly it hasn't been appreciated for too long. I think probably in the mid-90s is the first time I heard about it as a very new idea. And like a large organization that is accomplishing something, it may not be important the names of the people in that organization, but it's important what those folks do. You need somebody to handle the communication and somebody to do some logistics and those kind of things. And that's what goes on with the microbiota. So whereas the names and species of bacteria and parasites and so forth and the viruses present can be different from person to person, the core metabolome, what genetically that population of microorganisms can do, is a lot more constant. And that's what's on this picture here. I'm going to try to do something like a pointer, but I don't see it. Anyway, so you see on this panel, these are the different classes of microorganisms and how they vary with time. But you see how these 2 pictures over here are very much more similar. The different colors represent different metabolic capacities. So what we do with the microbiome is we do deep sequencing. We just do lots and lots of sequencing, and we can tell by who's there in the microbiome by these. That's okay. The tag of the ribosomal RNA, the length of it. So different parasites have 22 and bacteria have 16. We recognize this as a metabolically very active organ system, and I've had the opportunity working with Jeff Gordon for him to do some experiments in germ-free mouse models, which have been very insightful to us. There's the Human Microbiome Project. You might have heard about that. They had a series of publications in Nature last year, which is kind of like the Human Genome Project. I think many of you can recall that. Many of you weren't. Were probably in middle school or something when they had this going on. But we thought the Human Genome Project was really going to open the book. And it didn't automatically, really. And I think that's the same kind of thing with the Human Microbiome Project. Just vast amounts of sequence information and knowing who's there has not automatically led to advances in human health or so forth. Our hopes are, of course, always tend to be articulated, and this could become a new cure, a new drug, a new clinical intervention, or so forth about the microbiome. And this is the microbiome at work, and the dysfunctional microbiome you can see is recruiting, allowing macrophages and immune cells into the lumen, blocking IgA secretion, not making these short-chain fatty acids that are very important for not only fuel for the colonocytes, but as an immune modulator. And absorption of our food. We just don't appreciate, and you really can't put numbers and quantify on it yet, how much this matters. But I honestly believe, my opinion is, if we start looking into this in 5 or 10 years, this whole idea of, nutrient requirements and stuff is going to change a lot, because these microorganisms are making a lot of these vitamins and minerals for us, or freeing them up, putting them in a bioavailable form. So that's kind of a layman's tour of what the microbiome is and does. The population of microorganisms in the gut playing a key role in terms of making nutrients available, also being an immune modulator, something that varies over time. We know from what Marion said that, caloric intake is going up here in the U.S. And we know the microbiome in obese folks, which we'll talk about a little bit later, is different than folks that are of a normal body habitus or a thin body habitus. And we wonder which of those happens first, and whether there's some relation to obesity and changes in the microbiome that we could potentially manipulate for human health. So my first work with the microbiome and Jeff, we just did something where we looked at how the microbiome changes with age and geography. And we took some Malawian kids and Malawian adults and Amerindians, these people are in Venezuela, and some U.S. Twins, and took multiple samples of their stool and just cataloged who's there in there and how that changes with time. And we learned a few things. One is that this here is a variation from the adult configuration. So when children are first born, their configuration is much more simple than it is when they're adults, and it takes about 3 years for them to develop an adult configuration of the microbiome. Where you live matters. So these are principal coordinate axes, which are probably a little difficult to explain, but they have to do with the metabolic and genetic capacity of these populations. So very different among Americans. Malawians and Amerindians have more overlap. Amerindians are eating a lot more animal products. Malawians are eating almost entirely a plant-based diet. And yet quite a bit of similarity. The length of these bars, when the difference between the length of the bar isn't very much, things are more similar. And so there's more variation as children children are more similar to children in other parts of the world than they are to adults in their own communities. So this large cataloging exercise, we learned that, things are going to change over the first 3 years, so it's going to matter how old kids are and what their diet is in terms of when we collect these samples. That there's more interpersonal variation than adults. We are converging to a core microbiome, if you will. as we mature, that there's more bacterial diversity with age, and diet matters. So if you eat a lot of plants, you have a lot of bacteria that will digest those. Now, malnutrition, the picture I showed you to start with, it's not just that form, but in all forms, undernutrition, I guess, in all forms, can be defined as inadequate or excessive dietary consumption. Notice it doesn't say anything about the microbiome there, so that may be a definition that changes in some way, but still contributes to half of child deaths. And so child deaths have been dramatically decreased. The absolute number of child deaths has almost gone half since 1990, in 20 years or so. But still half of them are because of malnutrition. It's a pretty unexciting, sort of sexy kind of problem to have, to be so sick because you don't have enough to eat, then you die. But it is much more big problem than malaria, TB, and AIDS. And then we've seen some stuff shown this morning how obesity in the last 30 years is going through the roof. Okay. So how might the microbiome fit in with malnutrition? Well, you can imagine this cartoon just sort of highlights that. It may have to do with nutrient absorption. It may have to do with chronic gut inflammation, environmental enteropathy. Immune function, I wouldn't just say decreased, but dysfunctional. Recruiting different elements of the immune system which aren't necessarily needed or helpful. The gut microbiota protect us against enteropathogens. And so you can see if half of children are dying from malnutrition, the gut microbiota might be playing a pretty important role there. So let's get back to the child with kwashiorkor. So we look for, I've been working in Malawi since 94, so we spend probably 12 years examining a variety of different potential causes of kwashiorkor, and then we get ourselves to the microbiome to look at it and try to discern what role it might be playing in kwashiorkor, whether as the microbiota and the immune systems co-evolve, is there something dysregulated about these two in kwashiorkor? Is it not processing nutrients in the way that it should? We took 316 Malawian twin pairs, and we followed them for the first 3 years of life. We knew twins were at high risk for kwashiorkor just by taking care of these folks for the decade before. And we knew that it was pretty common that twins didn't always get kwashiorkor together. And so you might think, well, what is going on there? They live in the same house. They're exposed to essentially the same environment. They're fed by the same mother with the same kind of food. What is going on there? Why is 43% of these twins, one of them developed kwashiorkor, and only 7%, both of them did? And we took fecal samples all the time. They saw them every month in clinic, and we took fecal samples probably half of the visits they made. And whenever they got malnourished, we took lots more fecal samples and did dietary surveys, so we knew when their diets were changing in relation to this appearance of malnutrition, and took a look at the microbiome. So we were particularly looking at these discordant twins. What on earth is different from these folks that seem to have a pretty similar environment, and at least they share either half of their genes or all of their genes together? About 25% of them are monozygous twins. So if we look at these stools in the healthy co-twins, and then when children get kwashiorkor, they're fed ready to use therapeutic food, and we also give food to the twin because we don't want we want the sick child to have enough and not be shared. So what happens to their flora? Well, in terms of this amount of diversity, they're not that different. The quash twin from the KED, the healthy kit twin. But when they're fed this very rich diet, their diversity is going up, their microbiota is getting more healthy. Here, not quite so much. over here on this axis, we're talking about the relative abundance of one of the families of bacteria, Actinobacteria. And there's more of it in quash, and it seems to recover even with the therapeutic feeding, even though it does go down. So these aren't the same. They aren't the same pattern. They aren't the same temporal pattern. And we took these stool samples from the time that kwashiorkor presented on the child and their healthy twin, and we put them into a germ-free mouse. And we have some data to show that this is a very the population of microorganisms that grow up in that germ-free mouse is very similar to the ones that grow up in the humans. So it's not selecting out or something. And we put the healthy twins microbiota in, and we give them this bad Malawian diet, which what's bad about it is it has very little fat. It's missing many vitamins and minerals that people would say are essential. I would say they're essential. And, they putter along. They lose some weight. They're given a rich diet. They regain that, and they putter along. And then after a Malawian diet for a prolonged period of time, they just can't survive. The kids with the quash microbiota, well, they tolerate the Malawian diet a lot less, They lose weight and do clinically poorly a lot quicker than the kids with a healthy microbiota. Recover some with the food, and ultimately just having a bad diet like that is not a good thing. So those are remarkably different patterns. These here are samples from the humans. And so these are the healthy twins, and the lines go between like where, one time point in time sample and another time point sample. And the quash twins are less diverse. They're more mature in terms of their microbiome. When they're put on the same diet that's absolutely complete, if you will, these converge, but they don't overlap. You can see that these are kind of farther south than this one. And we rarely see children get quashed twice, but so looking at our mouse model, we tested those stools carefully for known pathogens. It was not that those stools contained some kind of known enteric pathogens. It was these commensal organisms that seemed to be having this effect. We have lots of information about 37 species level taxa differences, but a couple that might be a little more easy to understand are Biophilia wadsworthia is a pro-inflammatory bacteria. This is more prevalent in the quash specimens as well as this opportunistic pathogen. And then it just changes like overnight. It's so responsive to time, this microbiota in these mice. I mean, you might think it takes days for this to habituate and change. And 4 hours later, I mean, it's you feed them RUTF and it's completely different. So this is a very facile organ in our body that's changing really pretty quickly. And this, again, is just a cartoon of some of these findings. These kids, they're twins. They're eating the same diet. They follow them for 36 months. One of them gets quash, and we analyze that microbiota has reduced overall gene content. It's a more immature. It's not the same as their normal twin before they've had. And these are people who are unlikely to have many antibiotics. In the mice, we see those same kind of changes. We see the healthy twins stool leads to a microbiota that's more resistant to the poor diet that they're subject to. And the kwashiorkor microbiota is associated with very bad sort of clinical response from the mice. So I mentioned what I think was more important than the names of the bacteria in the microbiome, but the metabolome, what the metabolic capacity collectively is of this population. And these pictures here, just to sort of teleologically show you that, so quash would be running in this lane, and the healthy kid would be running in this lane. And yellow means there's more expression, and dark blue means there's less expression, and black is kind of a neutral color. These colors down here tell you what kinds of enzymatic and metabolic activity this is involved. So wow, these look very different. That's the first thing you'd say. These children don't have when we do a family phylum tree, they don't look as different as they sure do with this metabolic profiling. I mean, these kids with quash are making different amounts of different genes and processing different amounts of nutrients. And then you give them food, healthy diet. You can see this RUTF. These kids are doing all the right things with it in terms of making these nutrients available. The quash kids aren't using it quite as effectively. Instead, they're focused on some more basic survival functions. And then when we're done treating them, there's something intrinsic or innate about this microbiota, because they don't, even though they converge, they don't get to be the same. And looking at this, we see that in kwashiorkor, metabolism that's disrupted are sulfur-containing amino acids, cysteine, methionine, and taurine, also folate and B12. It's not necessarily a deficiency in these things, but there's something different about the metabolism. And obviously, I say this is like a first chapter in a long story about the microbiome, so I can't tell you the end of it. But I think this is one of the first times we've seen differences between children with quash and children without. So, and then I have one more of these pictures. I'm really proud of these. Their colors stand out and make it easier for you to understand. But on the Malawian diet, the red lines are the kids with quash, and the blue lines are the healthy, blue are the quash, and red are the healthy ones. And you can see that there are some differences between them. And then when you feed them the RUTF, the healthy ones, the red ones, the quash ones are lighting up more yellow. They're making more of these nutrients and metabolizing more of these nutrients because they need them. But if you look back at the red ones here, you'll see that these, you probably don't appreciate it, but there's more light in like that band than there is in that band over there. Remembering blue is the most negative. It's under expression. So these flora do not just completely revert. So, I'm a translational scientist. I'm a clinician, right? I'm not particularly able to conceive of the most advanced laboratory techniques or even the most advanced sort of statistical techniques. But one thing my students have said about me, and I've said this to a couple of people this morning, is they say I'm really unique because I can go toe-to-toe with both of these kind of groups and try to be a bridge of bringing new science knowledge to public health kinds of interventions. I think the kwashiorkor story is interesting in that we're seeing, we've done a lot of work, and these are, we have 15,000 stool samples from these cohort of 300 kids that we followed. And we have all kinds of information about what's going on with them. We've teased out the role that breastfeeding plays and the protective role and how that may be related to this. We found that these microflora are present there, these changes before the one child stays healthy and the other one gets quash. And hopefully sometime we'll be able to do something on a prevention level. So with that. And we'll talk about that at the very end. I wanted to share some data that's not mine, which is about obesity. We all know obesity is a huge problem. We know that there's phylum-level differences in the microbiota in obesity. There's reduced bacterial diversity in the obese stools from obese people. Jeff did a twin study where one twin is lean, one is obese. He had about 54 twins, some monozygotic, some dizygotic, young, healthy women. Nobody has lots of complicating chronic diseases. complete medical, lifestyle, and dietary data, and then just complete sequencing of their stools to find out who's there and what the metabolism is between those two. So here, if we look at these this number here is lower is more similar and bigger is more diverse. And you can see that there's not really much difference between monozygotic or zygotic. We don't think genetics is playing much of a role here. But if you look at the overall height of these two bars and these two bars, it's interesting that it's more diverse, not interesting, but you might expect, and more diverse that between generations, twin to mom and twin to twin. If you look at the amount of phylogenetic diversity, the lean folks have it over the obese folks. And in fact, there's the odds ratio of about 400 or so genes was different between obese and lean twins, and most of these genes were coming or expressed in these bacteria, Actinobacteria and Bacteroides families. So, I mean, this is all kind of exciting. It's new science. You know, we've just been opening up this technology of deep sequencing and how that's going to perhaps, open our eyes to what's causing kwashiorkor in kids. Maybe it's going to do something for obesity. And we can be very hopeful in some ways about future research. And as I said, the immune system, the role of antibiotics, and how that changes the microbiome are potentially areas where new knowledge is going to come in pretty quick. But this is a place that's called a center for policy. So I want to finish here by talking a little bit about the future of public policy with the microbiome, because it's nowhere, as far as I can tell, on the radar screen. And the way, in broad strokes, the way we're going to change the microbiome is through foods and diet. We're not going to do it it doesn't really work with stool transplants or probiotics. We have a bunch of other data to show that these are very bad ways to try to change the microbiome. And very good ways are to feed people different foods. And how do with food products, do we ever require data on the effect of the microbiome when new food products are introduced? We live in a nation that, as far as I can tell, shuns cooperative governance and devalues public welfare. So we have to be a little bit forward-thinking and careful about when people introduce things, how that might change the microbiome and how that might change health. And this is not that the microbiome is the answer to everything, but it's a player that you can see in there. It's a sixth organ system. And do we need some kind of policy structures to evaluate foods nationally and internationally? What are the risks of altering the microbiome? I mean, if somebody had some sort of herbal starch product out there on the market, it wouldn't be regulated very much. It might do all kinds of bad things to your microbiome and might make you much more susceptible to infection. I mean, do we have to learn about that by sort of trial and error and total experience? So I think this is an area that really, I would hope one of these policy places like Berkeley would think a little proactively about and so forth. So that's really about all I have to say about the microbiome. I think it's important. I think it's definitely an under-recognized player. I think we don't know hardly anything about it. I don't even think we know 1% of what we need to know about it, how it affects human health. But we also need to get our thinking, as Marion pointed out, in a system that goes all the way from, she would go from agriculture to health. I mean, I would go from cellular biology to population biology or health, across those sort of areas. So I've said enough. Thanks.